Thank you very much, Alice. Thank you for the kind introduction. Yeah, so I am, just to say, I'm also at the Quadram Institute and also affiliated with the University of East Anglia. Yeah, but principally I'm at the Earlham Institute. So today I'm going to talk about genome-resolved metagenomics, and by that I mean the methodologies that we have for resolving microbial genomes direct from metagenome data. And this has been quite revolutionary over the last five to 10 years. It's actually nicely illustrated by this tree from Hugo et al. In 2016. So this tree shows all the bacterial and archaeal phyla, and also the eukaryotes down there, known at that point in time. And then every red dot corresponds to a phylum for which there are no cultured representatives. So no microorganisms from that group has been successfully isolated and sequenced. So the question then is, how do we know about the existence of these groups? And that is principally through what I'm going to talk about today, genome-resolved metagenomics. Without the necessity to actually grow these organisms, we can extract their genomes from metagenome data. And I'm going to talk about, in particular, the case when you have multiple metagenome samples forming a time series. The metagenome being the mixture of genomes of all the different microorganisms in that community. And as I said, this has been quite radical in terms of transforming our understanding of environmental microbiodiversity. We found all sorts of wonderful novel organisms. A good example is this candidate phyloradiation, which is this diverse group of organisms with minimal metabolisms found through the work of Jill Banfield and others. But also it's been really important in the context of the human microbiome. So in a recent Nature review, it was pointed out that one of the major milestones in the study of the human microbiome, so that's the host-associated microbiome of human beings, and I'll talk about that a little bit more later, was the generation of these large-scale gene catalogs. And they were also obtained through metagenomic binning and the extraction of genomes directly from metagenomes. Okay, so an overview of my talk. I'll talk a little bit, I'll give a high-level overview of the algorithmic background to metagenome binning, but without complicated details. I'll illustrate this with an example from my own research. We're actually looking at a system called an anaerobic digester, which I'll talk a little bit more in detail about. And then I'll discuss algorithms that you can use to actually find subpopulations within species. So go to the level of strains in your microbial community. Again, in this context where you've got a time series of multiple samples. So this is just giving a brief overview of metagenomics bioinformatics. So as I mentioned in the introduction, the starting point for metagenomics is when you have a whole community of microbes. So we've got all these different species present. And the key thing is that they're present in different abundances in that community. We would extract out the DNA and we sequence it directly. So this is, without the need, as I said, for isolation or culturing of those organisms. And then all the methodology I'm going to talk about today basically corresponds to this part of the diagram here. I'm going to talk about assembly-based methods for metagenomics. But the first thing you do with this, all these short read sequences is you try and assemble them together, and then I'll talk about how we can bin them into the genomes present. There are a whole group of methods where you work directly from the reads to get taxonomic abundances, functional profiles, etc. And those are immensely useful to give you an overview of a community, but that's not what I'm going to be focusing on here. So just very briefly, what is assembly? So assembly is essentially, it's a very simple process. So we've got these multiple reads from these different organisms, and they're short. So typically short read sequences, there might be a couple of hundred base pairs. So the first thing we want to do is look for overlaps between one read and another. And here I'm showing a very simple example. We've got these two reads. We can make a longer contig. The challenge arises because of repeat regions in genomes. So if you have a identical repeat region within a genome that exceeds the length of your read, then you have uncertainty in the assembly process. We have a read that starts here and runs into this version of the repeat, and another read that runs out. But then we have a third read that comes out of the second version of the repeat. So when we come to assemble, we can overlap this with either that read or that read. So we have a sort of natural uncertainty in the underlying sequence that is explained by the set of reads that we have. And we represent that uncertainty with something called an assembly graph. Now, I'll be showing quite a few assembly graph diagrams, so it's useful to understand how they work. And so an assembly graph would be produced whatever method you've used to actually assemble your sequences. If you've used de Bruijn graphs or string graphs, you'll still get an assembly graph. And as I said, it represents the fundamental uncertainty that exists within your assembly, given the reads that you observe. And then each path in an assembly graph corresponds to a different possible read. Each node is an unambiguous sequence, and then the edges correspond to overlaps. There is a little bit of subtlety in terms of you can enter and leave those nodes either on a reverse complement or not, but I won't go into that. So we have a sequence here. So I sort of made it very trivially all A's, and then we have two alternative sequences, two alternative paths, generating possible variation that are consistent with our reads. One version has a G, the other version has a T. And it really is that simple, except for when you start looking at real graphs. So this is an example from a metagenomics example. I've got 6 different strains of Lactococcus lactis, and I've built I've generated random synthetic reads from those 6 strains, and then I've built a synthetic graph, and this is based off short reads, 2 by 150 base pairs, which is the typical high throughput technology at the minute. And the key thing is you can see that there's a huge amount of uncertainty in this graph, and that arises because of repeat regions within genomes, but it also arises from the particular complexity of metagenomics, and that is that you will have repeat regions between genomes. So if you have 2 very similar strains, part of their genomes will be shared. So we get a lot of, so we get a highly complicated graph with this potentially tangled structure. So there are actually 6 paths through here, but it would be very hard to resolve those paths. So instead, what we do is we take the short regions of this graph, which are unambiguous, and we call these contigs. And we work directly off those contigs and try to link them back together into the genomes that they came from. So this is Contig Binning. And essentially, in Contig Binning, we use 2 features of the contigs to try and cluster them back together into the genomes. The first feature is sequence composition, which we very simply obtain by looking at k-mer frequencies. Typically, actually, for this problem, we would use tetramers, so k-mers of length 4, but I'm illustrating this with k-mers of length 2. So all you do to get the sequence composition is literally count up the frequency of different unique k-mers. So we have a sequence here. It's got 2 ACs in it, so it gets a frequency of 2 for AC. For the CA, frequency of 1, etc. So in that way, we can represent a sequence in a relatively low dimensional space, which is the frequency of these unique k-mers. And the key thing is then that all the sequence derived from the same genome will have a characteristic set of k-mer frequencies. And I'm showing this here from an example where I've taken 5 different species of bacteria. I've generated 10 kilobase pair fragments of sequence. I've counted up the frequency of unique tetramers, and then I've log transformed them and done a principal components analysis to reduce the dimensionality to 2. And you can see that we get these distinct clusters in the sequence composition space. Well, actually, the key point is here that they're diffuse clusters. So there is a significant amount of overlap, at least when we look in the first 2 PCA dimensions. So, because we're, so an extra source of information that we can then use to more reliably then cluster those contigs back into the genomes is something we'd call differential coverage depth. And the key principle here is that we start our assembly not from just one sample, but we do a co-assembly of multiple samples. So this is where we get the longitudinal time series that we're focusing on on this lecture. So we generate a co-assembly, contigs from co-assembly, and I'm showing two examples here. And then we map the reads back from each sample separately to those contigs. And then we compute something called the coverage depth, which is basically just the average number of reads spanning any point on that contig. And you can figure out that that will be given by this simple formula here for a particular sample. The number of reads mapping onto the contig multiplied by the length of the read divided by the length of the contig will give you that average read depth. So we compute that for each contig in each sample separately. And here's a little example from this paper by Albertson et al. In 2014. They actually had two samples, which they'd generated from different extraction methodologies. And then they've done a co-assembly. They've got the coverage depth in each of those samples. And you can see that you get distinct clusters. What is happening here is that all the contigs that derive from the same genome will have the same profile of coverages across the multiple samples. And that's because they all that genome will have a particular set of copy numbers across those samples, which then relates back to this coverage depth. So this is an extra form of information that we can then use to bin the contigs back into the genomes they derive from. And this is basically the strategy used by all modern contig binning algorithms. So an example would be, say, Anvio, which is a manual binning algorithm. There they use their composition and coverage is used together with a hierarchical clustering algorithm, and then you perform manual binning. Hierarchical clustering, though, doesn't scale very well. So most automatic contig binning algorithms will be based on some form of partitional clustering, where you're essentially trying to group the contigs into a certain number of groups. And the classic example of partitional clustering would be the k-means algorithm. K-means is very simple. You basically choose 3 initial random clustering centers, and then you reassign each data point to the closest cluster center. You repeat, and then you compute the mean of the cluster. So you recompute the mean, and then you reassign the data points, and it's guaranteed to converge after generally a fairly small number of iterations. So if you can devise an appropriate distance metric that takes into composition, the composition and coverage of the contigs, you can very easily apply these types of partitional clustering algorithms to actually do your contig binning. And k-means, as it turns out, isn't actually a bad algorithm for this problem. It's something similar was used in the first version of MetaBat. Since then, people have developed increasingly complicated binning algorithms, which address, so they have more complicated models for the coverage or the sequence composition, and also address the question of how you define how many clusters or genomes are actually in a data set. I'm going to talk a little bit in more detail about the algorithm we developed, Concoct, and this does differ at least from the early algorithms in this area, in that it has an underlying probabilistic model to the actual clustering rather than being simply based on a distance metric. Okay, so in Concoct, you first perform the data pre-processing. That would consist of this co-assembly across the multiple samples. Then we fragment contigs that are greater than 10 kilobase pairs. We do that for 2 reasons, essentially because long contigs are potentially chimeric, but also because it adds more statistical weight to long contigs if we fragment them. We generate this Kamer frequency for those contigs. We normalize the coverage, and then we just simply join those two vectors together, and then perform a PCA on those joined vectors. And the point of the PCA is not for visualization, it's to reduce the dimensionality of the problem. So with tetramers, you'll have a dimensionality which is 133, because there are 133 unique tetramers taking account of reverse complements and palindromes, etc., plus then the number of samples that you have. So we perform a PCA and we keep enough dimensions to maintain 90% of the variant. And then the actual clustering algorithm is just a form of Gaussian mixture model. So in a Gaussian mixture model, we're representing a more complex distribution, which in this case is our probability distribution across the different genomes or contig bins, and we're representing each cluster as a Gaussian. And we're using a completely arbitrary Gaussian, which basically corresponds to fitting arbitrary orientation, arbitrary size ellipses to each of our clusters. Now, we use a methodology called variational Bayes to perform the actual fitting, which is an approximate form of Bayesian inference. And in this context, it's useful because it means that we can use automatic relevance determination to determine the actual number of clusters that are appropriate for any given data set, rather than having to predetermine them. And what happens in automatic relevance determination is essentially you start with too many clusters, and then the ones that you don't need will be shrunk out of the solution. So Concoct we developed back in 2014, but Contig binning is now essentially a very crowded field, probably because it's relatively easy to devise clustering algorithms. Some of the most popular ones are MetaBat2 and MaxBin2. More recently, VAM came out. That was interesting because it used a variational autoencoder for the pre-processing step. Concoct itself was used in some very significant discoveries in microbial ecology, for instance, the Asgard archaea and Comomox. And it's actually still fairly competitive as a binner. It probably doesn't do quite so well in terms of the maximum number of good prokaryotic bins. But if you're not just interested in prokaryotes, so for example, eukaryotes and viruses, because it makes no assumptions about the nature of the bins, it can give you a sort of nice overview of the entire community. Most of these algorithms, though, are still based on the same information as Concoct, a combination of coverage and composition. More recently, some nice algorithms have appeared that utilize the assembly graph in addition to coverage and composition to improve on the binning. Okay, so that's how we do contig binning. That gives us clusters of contigs. Then the next question is, what are those clusters of contigs? They could correspond to viruses, as I said, they could correspond to eukaryotes, but in general, what we're most interested in finding are prokaryotic genomes. So how do we decide whether a cluster is a good prokaryotic genome? We essentially do that in a very simple way. We look for something called single copy core genes. So these are genes which we know should be present in every prokaryotic genome and present in a single copy. And it's that single copy core nature which is important, because then we can look at our bins, and if we see these genes all present in a single copy in that bin, then we know it's a good potential prokaryotic genome. And this is the principle used by, for instance, the CheckM program for evaluating bins. And it will give you statistics like completion, which will be the fraction of single copy core genes that are present, and contamination, the fraction that are in multiple copies, which gives you an indication of whether there are more than one genome present in a particular bin. And then metagenome assembled genomes are basically just contig bins that satisfy a particular criteria in terms of completion and contamination. Okay, so that was a brief overview of the algorithms. Now I'm going to give you an example from one of our recent analyses. This is available on the bioRxiv if you're interested. And this was a longitudinal analysis of a set of anaerobic digesters. So anaerobic digesters are a green biotechnology that convert organic wastes into methane-containing biogas. And it's green because it both treats that waste, and then you can use the biogas to generate heat and electricity, rather than wasting that resource effectively. So we performed a survey of in total 12 different anaerobic digesters around the UK, and we got weekly samples from each of these digesters delivered to us on ice. And then we performed a co-assembly of each of the 13 anaerobic digesters separately. So we had 390 samples in total. We co-assembled with MegaHit, which is about the most efficient short read assembly algorithm. So then we've got 13 co-assemblies, one for each digester. We did concoct to generate in total 17,000 bins. And then we did a preliminary filtering based on those single copy core genes to see which of those bins look like they might be good mags. Now, there is a subtlety to the mag evaluation that I didn't really talk about, and that's that probably for different large-scale groups of organisms, you want to use different sets of genes. So having got these sort of possibly good mags, we then assigned a taxonomy to them using a program called GTDB TK, which is a really neat program that contains 100,000 or so mags and genomes in a nice tree, which gives you an efficient way for assigning a taxonomy off those single copy core genes. And then we group them into bacteria, archaea, and candidate phyla. So these are this novel group of bacteria which have sort of a quite distinct genomes. And then we used a different single copy core gene panel for each of those three groups to then refine our mags into a set of some 5,000 that look like good quality mags once we've done this refinement on phylogeny. And then because we want to compare across reactors, we used another program called DREP to dereplicate those mags across our 13 reactors so that we can look for commonality in microbial diversity between the different anaerobic digestion reactors. Well, this is just showing across the whole set of 2,000 mags, their quality in terms of those two statistics I mentioned, the completion of single copy core genes and the contamination. So down here, we have really high quality genomes, things that are almost entirely complete and not contaminated. Whereas I suppose over here, we have the opposite. These would be, well, these are things we didn't count as mags basically. But yeah, you've got on the y-axis increasing levels of contamination, and then on the x-axis going that way, decreasing levels of completion. Okay, so that gives us the diversity in the reactors. But another key aspect to this type of longitudinal analysis is the actual abundance of those mags over time and between communities, because that's what we can use to actually look at the ecology of these microbial communities. So how do we get that abundance? Now, if you've done a co-assembly in one reactor, you can obviously take the coverage depth for each for that mag in the reactor where you have assembled it. You would normalize it in some way, either by total read number or coverage on those single copy core genes, and that will give you a coverage profile for the mag in the reactor that you found it in. But that mag may actually be in other communities, but for various reasons failed to actually be assembled or binned. It may have been at too low a coverage to be successfully assembled, or it may be because of strain diversity, as I'll talk about a bit later. It may not have been successfully binned. So to sort of try and pull in those missing mags, we also mapped the contigs from unmagged, which weren't successfully magged in a particular reactor, back onto our bin collection using Minimap2, and that enables us to sort of augment the coverage of each mag. So now we have the coverage of a mag in the reactors that it was binned in, but also in any reactors where it may have been there but wasn't successfully constructed. Okay, so this is just summarizing, this is providing a phylogenetic tree of the 2000 mags that we obtained. So you can see we've got a whole lot of diversity, some of it including substantially novel groups. I think we've got novelty at the order level in this study of anaerobic digesters. One thing that's quite neat actually is that most of that diversity is in the firmicutes, which would be similar to the human gut. In fact, the well-studied organisms, like say, for example, the proteobacteria, actually form a relatively small fraction of the true diversity in the community once we can use these methods to properly evaluate it. As I said, we're not just interested in the diversity, we're also interested in using the abundance of those mags to understand the community dynamics. And this is what I'm showing here for each of the each of 12 reactors for which we had a decent time series. I'm using UMAP to map down the mag coverages to 2 dimensions so I can summarize the community profiles over time. What's very noticeable is that in most reactors, we see a clear shift in community structure over time. So AD 2.1 here, for instance, we start at this point. So each point is a different sample. So we can see that over time, we start here, and by the end of the year, we've got a completely different community structure. And what's was very striking to me is that is true for the majority of the reactors, in fact. So AD 11 over here, we can see again, there's this clear shift. There's a couple of them where there might be something that looks like cyclic dynamics. So that's possibly true in AD 20 over here. But in any case, we can use this methodology to show that there's these clear changes in community structure. So we want to try and understand what drives those changes. And for me, the answer to that lies in actually trying to understand the function of these different mags. Now, an anaerobic digester is a complex community where you have many different types of microbes performing very different metabolic functions, but the overall process ends up with methanogenesis. So this is what I'm demonstrating here. And there's 2 essentially different pathways for that methanogenesis. There's something we call autotrophic, which starts from hydrogen and carbon dioxide, and something called acetoclastic, where you start from acetate to produce the methane. And this is performed by archaea, and it's relatively easy to understand. It turns out that the methanogens, the archaea that perform this process, are quite low diversity, but they also depend on bacterial syntrophs to actually produce the substrate that they use for the methanogenesis. And typically that's considered to be a very intimate relationship because it's generally very unenergetically favorable for the syntroph to produce the acetate or the hydrogen required by the methanogen. So the syntroph needs the methanogen to be there to keep the concentration of its product low, basically. So syntrophy comes from the Greek or Latin for sort of, together eating, basically. So the key point here is that the syntroph is providing the substrate to the methanogen, and then you have a whole complex set of processes performed elsewhere in the community, hydrolysis and digestion into monomers. So the question is, how can we assign the mags to these different functions? And here we utilized a method that was first proposed actually in the context of genomes by Alice McCarty's group. So they used a machine learning method to take a set of genomes with known traits. I think they were focusing on more sort of clinically relevant traits. And then they were able to treat each genome as a bag of genes and infer a machine learning model that enabled you to predict the trait of those genomes. I think they were using a support vector machine to predict the traits from the genomes. So we've done something similar, but for predicting the traits of our mags. And we used a different database of traits. We used one that was maybe more appropriate to environmental organisms, and we published that a few years ago on the bioRxiv. For this AD analysis, we generated a new model specifically for the methanogens, which enables us to predict for each of our MAGs which methanogenesis substrates a MAG is using. So we had models for 5 different substrates: formate, hydrogen, acetate, methanol, and methylamine. And here, for each of our methanogen MAGs, I'm showing the machine learning model predictions for the substrate utilizations together with a phylogenetic tree. So you can immediately see there's a very strong association with phylogeny of the trait utilization. Down here, for instance, we've got the Methanosuccinia, which are generalist methanogens, able to perform both of those pathways, the acetate utilization, which is the acetoclastic pathway, and the H2 utilization, which is the autotrophic pathway I was talking about. Whereas up here, we've got a diverse group of methanomicrobia, which are principally autotrophic methanogens. So we can use these strategies to tell us more about what the organisms in our reactors are actually doing. And then on the right here, I'm summarizing the abundance of those organisms across reactors. And then, as I was saying, the actual sort of community structure of the methanogens is rather simple. So we can see, for instance, that there's a whole set of reactors here that are essentially dominated by one of these Methanosuccinia, and then another set that are dominated by a different Methanosuccinia MAG. And we can take the same trait approach, but then summarize it at the whole community level. So we've got the trait assignments for each MAG, but we've also got the coverages of those MAGs. So then we can compute the overall abundances of methanogens capable of performing these different traits. And when we do that, we can see that the shifts in community structure are associated with a shift in methanogenesis traits. So that is possibly one of the things that might be driving the shifts in community structure that we observe. The other thing that we were very interested in was trying to identify the syntrophic repairs in our reactors. And what we've, from looking at the abundance of potential syntrophs and methanogens, we could see incredibly strong correlations between some pairs of organisms in terms of their abundance. So this is what I'm showing here. We've got a methanogen MAG. I think it's the red line, and then a non-methanogen, the blue line. And you can see how closely the abundance of those two organisms are tracking each other. And here I'm showing it as a correlation plot. So on the x-axis, one MAG abundance, on the y-axis, the other. So what we proposed is that these very strong correlations may be possible syntrophies between the methanogens and the organisms provided their substrates, and to sort of to determine the usefulness of that hypothesis, we looked at whether the potential syntrophs had a different set of genes to the rest of the community, and we could actually see that they are upregulated for things like sugar transport, but also oxidoreductases, which are sort of classic of examples of what a syntroph might need for passing things like formate or hydrogen to a methanogen. And again, we could actually build a machine learning model from the KEGG orthologs in a MAG that is capable of quite accurately predicting whether it is whether it will correlate with a methanogen or not. So that gives us confidence that these things we're seeing in terms of abundance might actually reflect something of the real metabolism of the organisms. Okay, so that sort of concludes the first half of my lecture, really. That was all about MAGs and how we can extract them from metagenomes in these longitudinal contexts. I'm now going to go on and talk about how, again, using these patterns of abundance across multiple samples in a time series, we can resolve strains, so not just the species in our community, but the subpopulations. So what do we mean by a strain? Well, this was, there's a nice review paper from 2005 where across the genomes that were available at that point in time, they compared average nucleotide identity against the total DNA sequence that differs between pairs of genomes, both within a species and between species. And a strain corresponds to this group here, where we're doing the comparisons within species. We've got things that are more than, less than 95% different. But the key thing here is that things can be very similar in terms of sequence identity in microbes, but can differ a great deal in terms of overall genome content. So these E. coli, in fact, that are differing at just a couple of percent nucleotide identity have an entire megabase pair different at the genome level. So it's these strains that we're now going to try and resolve again de novo from the metagenomes. And that they can be very important functionally. Again, the classic example would be E. coli, where one strain can be a harmless commensal, and another strain will be a pathogen that can make you very sick. So inside these mags, then we have subpopulations, and there'll be various processes defining those subpopulations. So clonal expansions, niche selection, they will all sort of maintain the subpopulations, but then you'll have recombination between strains, which will serve to dilute it. If you have a well-studied organism, so an example might be Salmonella, for which there's over 200,000 genomes available, then you can use a reference-based approach for resolving strains. But in general, as I said at the beginning, the majority of organisms are either hard to culture or haven't been cultured in any great number. So this motivates the need for a de novo approach capable of resolving strains straight out of metagenomes. Okay, so the variation between strains in a metagenome will consist of two forms. One will be nucleotide variation on shared genes, and I'm illustrating this here. So we've got a contig where there are two different variants, and those variants have different abundance across samples. So in the green sample here, we've got the A variant. In the red sample, we have the C variant. But we'll also have variation on the accessory genome. So these will be genes that are particular to one strain and not found in another. And that corresponds to that difference in the genome structure that I was talking about in the last slide. And there'll be two sources for that variation. One will be essentially population genetics. And that isn't going to be the sort of target of the algorithms I'm talking about here. But the other will be actually distinct clonal subpopulations. And that's what we'll aim to resolve from the time series data. I've been working on this now for quite a few years, and I'm going to talk about the first version of our algorithm, Desmond, and then I'll go on and talk about the second version, Strong. So Desmond uses the same principle as we were using for clustering contigs into bins, and that's co-occurrence across multiple samples. But it becomes a little bit more complicated when you have strains for the reason that I'll explain. So here I'm illustrating the principle. So this is taking a single gene where we've got 5 different strains present across a set of 64 samples. So each column here corresponds to a different sample, and then each row is a variant, a SNP on that gene. And I'm showing in the heat map the abundance of the relative frequency of the minor variant. So all these variants here, they have the same profile of abundance across samples. And we can use that to link them into the haplotype that they derive from. Similarly, here we have another set of SNPs that all derive from the same strain. So this is similar to the clustering of contig bins. But the essential difference is that we have some profiles that are a summation of the two underlying strain profiles, and that's because both of those SNPs were shared in those two strains. So the profile here is then the summation of the two underlying strain profiles. So it becomes a deconvolution problem rather than a simple clustering problem. So we implemented this in a pipeline called Desmond. We published it in 2017. The basic principle is we take different samples, generate the contigs, get the bins, and then we look at those single copy core genes again on the bins. But now we find the frequencies of SNPs on the single copy core genes, and we're able to deconvolve the strains present, their abundances, but also their haplotypes on those shared genes. As always, it's difficult to decide on the actual number of haplotypes present. We use a metric called deviance to determine that. Basically, you increase the number of haplotypes until the deviance stops decreasing, and you determine that to be the number of strains present. And then we have an additional step that enables you to estimate the accessory genome for each of those strains using the non-core gene coverages across the multiple samples, essentially. All right, so I'm just going to go and show you one quick example of Desmond before being applied to a real data set before I go on and talk about our more recent algorithm. So this was an application of Desmond to a set of mags generated from the Tara Ocean Survey. So this was, Tara was where they sent it still is where they go around the entire world sampling metagenomes from multiple plankton samples. And Tom Delmont, my collaborator on this, did a large-scale binning across Tara and identified some 660 mags. I took a subset of those, the ones with high coverage, basically all 32 mags that had a coverage greater than 100. And then I looked at these single copy core genes and looked at the variant positions. And actually just doing that, we could see an interesting relationship that there was a negative correlation between the number of KEGG pathway modules in a mag and the overall number of SNPs on those core genes. So basically, as things get more metabolically complicated, we're seeing less nucleotide diversity within a mag. And then we use this Desmond program to actually break that diversity down into the underlying haplotypes. And I'm showing one example here from over the 30 mags. Actually, most of them did have strain diversity. And I'm just showing one example from a proteobacteria, a highly streamlined organism, which we were able to deconvolve into 4 different haplotypes. And the key thing is that we could see different patterns of geographical abundance in those haplotypes. So one haplotype was found in the Mediterranean, another was found in the mostly in the Pacific Ocean, and the third one here is in the Indian Ocean. So this suggests that these subpopulations, these strains, may have some ecological relevance because they have distinct patterns of geographical abundance. So looking at all 30 mags that we're able to deconvolve into strains, we saw a significant negative correlation between the number of haplotypes in a mag and the genome length. So this is similar to what I was showing with the metabolic complexity. And then the majority of those strains actually did have a significant correlation with geographic region. And then the final thing we looked at was the accessory genome. So for each pair of strains in a mag, I've compared the nucleotide divergence with the genome divergence inferred from that last step of the Desmond pipeline, where you get the accessory genes. Now, as you might expect, there's a positive correlation between the two things. As two strains become more dissimilar on the core genes, then they're more likely to diverge in the rest of the genome. But what's neat is we actually saw a different relationship for the large genome organisms versus the small genome organisms. So the red line here corresponds to those mags which had a genome larger than 1 megabase pair, and the blue line, those that had a genome smaller than 1 megabase pair. And you can see that there's a stronger relationship between overall genome divergence and ANI for those small genomes. And that, I mean, and actually this was sort of discussed in this paper in 2005, basically how this gene content divergence corresponds to different ecology. So what this means is that a small genome has to diverge its genome more to potentially shift into a different niche, as opposed to a large genome organism, which may be able to shift into a different niche more in terms of changes in gene expression. At least that was our hypothesis. Okay, so I just wanted to give you a brief overview of Desmond, because some of the ideas are pertinent to the last program I'm going to talk about today, which is an algorithm that we call strong. So the principle of strong is again addressing this question of how to resolve strains in metagenomes. But now we're going to go back to the original assembly graph, because if you remember at the start of my talk, I said that the assembly graph is our way of representing uncertainty in the sequences that we have. So in essence, all that strain structure is present in the original assembly graph, but it gets simplified and cleaned out basically before during the process of forming contigs. So we asked whether it would be possible to actually resolve those strains directly from the assembly graph, and that would have a lot of advantages. We can avoid mapping, we can represent more complex variant structure, so we can have gene insertions as well as SNPs, and it also enables you to incorporate larger scale read information than looking at single SNPs as we do in Desmond. This was pointed out in spacecraft cats that this would be sort of a useful way to look at strains, but ours, I believe, is the first practical algorithm for prokaryotes. At least there was a similar sort of thing proposed for viruses that actually utilizes the assembly graph to find strains. So how does it work? We actually have a similar principle to Desmond, in that we're looking at the coverage across multiple samples, again, these time series, but now we're trying to find paths on assembly graphs that are consistent with those observed coverages. So our input data would be a single copy core gene graph, and these are often actually very simple. Unlike the overall graph, which becomes very complicated, if you focus on the single copy core genes, they can be pretty simple graphs. So here is an example. We've got the unitigs here, and each strain corresponds to a different path through that graph. And we also have for each unitig, so there's unitig 9 corresponding to 9 in the heat map, we have the coverage of those unitigs across the multiple samples. So we're aiming to find the possible paths through that graph and the coverages of the strains across samples, which then deconvolve back into our original observed data. How do we do that in terms of a practical algorithm? Actually, we use variational inference again, as we did in Concoct. But now it's a much more complicated variational inference algorithm, because we're having to find paths that are constrained by the graph structure. So it's actually a form of structured variational inference. We update the paths for each strain in turn, then update the abundances of each of the different strains. So we actually formulate this as a conjugate Bayesian model, and then in practice the fitting consists of finding the possible paths, and one of the nice features is that the path is defined in a probabilistic sense. So we don't have a definite sequence for each strain. We have a whole set of possible sequences that are consistent with it, and then we update the abundances of each strain in the sample, and we've also got measures of noise, etc. And we also actually have to have some biases. It's a pretty complicated model. We iterate that just like an EM algorithm until we converge on the answer. And we use, again, the same principle as Concoct. We use automatic relevance determination. So we start with far too many strains, and the ones that we don't need drop out from the solution. We implemented this in the strong pipeline. So this is much like Desmond again. We start from multiple samples, generate contigs, build mags. But now the key thing is we store a high resolution version of the assembly graph prior to the formation of the contigs. And then when we've found the single copy core genes, we extract out those subgraphs from the high resolution assembly graph. And then when we've got the set of subgraphs for a MAG, we can resolve the number of strains that are present, their abundances, and their paths. So I'm going to show you a couple of examples of strong. The first, a very simple example, is actually from a fecal microbiome transplant. So generally in the human gut, most species aren't present in multiple strains, but the one situation where you will have a lot of species present in more than one strain is when you perform a fecal microbiome transplant. So this is a medical procedure that's actually a very effective way to treat diseases like C. difficile, and that's the context in which it was applied here. We actually use a colonoscopy to transfer feces from a donor into a patient recipient. So we've taken two individuals, the recipient and the donor, and we have a time series of samples from each. And then we did this co-assembly, computed coverages, and ran everything through the strong pipeline. This was actually from a study performed by Merrin and Andrea Watson as part of a much larger study where they've got some very interesting hypotheses about diversity and IBD. So I encourage you to go and look at the archive paper of that. But anyway, we used it as an example of how to use strong. So we've got 7 samples from the donor and 7 from the recipient. And we have 2 samples from the recipient pre-FMT and 5 post-FMT. And strong predicted 109 MAGs from these 2 individuals. And here I'm showing a heat map of the abundance of those MAGs across samples. So you can see that the recipient before the FMT had a very simple community, essentially just 3 different genomes present. There were some other genomes that I haven't put in this heat map because they weren't abundant enough across multiple samples. But we're just going to focus on one MAG here. And this one is interesting because it's present in the donor and it's present in the recipient pre-FMT, and then it is maintained after FMT. So here we have some potential for competition dynamics between strains. And effectively, that's exactly what we predicted from strong. So now I'm showing the time series of 2 strains. So for this MAG, strong predicted that 2 strains would be present. One is the donor strain, shown in red, and the other is the recipient strain, which was entirely present, which is dominating the recipient community prior to the FMT. Post-FMT, it's replaced by the donor strain. But what's quite nice in this example is that then you can use our algorithm to actually show that the recipient strain starts to come back again. So even though if you're just looking at the species level, you might think that this FMT has been successful. At the strain level, that you can see that there's more complex dynamics. Okay, so final example then is actually going back to those anaerobic digesters, the same ones we generated all those MAGs from. But now I'm just going to look at the strain dynamics in one digester. We took 10 samples, and here I'm showing the MAG abundances over time. So as in the larger study, we can see a shift in the community structure in terms of the MAGs, of which we got 309 good MAGs in this subset of the data. When we applied strong to all those MAGs, we were able to resolve strains in something like 40% of them. Here I'm just showing the MAG coverage on the x-axis against the number of strains predicted. So the two things that are positively associated, as you'd sort of expect. As you've got more coverage, you can pick up more of this strain diversity. And here I'm actually just showing one particular MAG. So, and here is one gene from that MAG. In fact, it's one of the larger genes that we look at. I think it's COG 55532. So for this particular MAG, we resolved 5 different strains corresponding to these 5 different paths through this graph. And you should remember that there are another 35 or so graphs, each of which we resolved the paths through, but I'm just showing one for convenience. And then we also get the abundance over time of each of those 5 different strains. And this is the overall normalized MAG abundance. But you can see something quite interesting from this, in that the MAG starts off at relatively low overall abundance in the community and increases to something like 4 or 5 times, but the strain abundances don't vary that much. And this is something that we saw across much of the community, in fact. So here I've summarized across all the MAGs that we resolve strains for. I've got the phylogenetic tree here, their abundances over time. And the ones I've highlighted in red are the ones where the MAG coverage went up significantly, and in blue are the ones where the MAG coverage went down significantly. But only for a relatively smaller number did we see a change in the relative proportions of the different strains. So it seems that the strain dynamics are relatively stable compared to the species dynamics. So just to conclude then, everything I've talked about here was based on short reads. As you get longer reads, a lot of those issues with repeats disappear, basically. A lot of them are caused by repeat regions that are shorter than your read length. As you get a longer read length, it becomes far easier to resolve strains. And that's something we're sort of actively working on at the minute. But in the context of strong, we simply use nanopore reads to validate the predictions from the short read strain resolution. So on one of the samples, we ran a single nanopore min-ion flow cell, got some 40 gigabase pairs of reads, and we looked at the MAG that was most abundant, bin72, and strong predicted 2 haplotypes for this MAG. I then mapped all the nanopore reads back onto that gene and sort of calculated a sequence distance to the haplotypes, and again plotted it into 2 dimensions. And you can see that we do get 2 distinct clusters of nanopore reads consistent with there being 2 underlying haplotypes that we predicted from the short reads. And actually the proportions were very similar too, which to me was super interesting because we've used entirely different DNA extraction protocols for the long and the short reads. So it seems that DNA extraction protocols, which may strongly bias species proportions, don't impact strain proportions so much. But anyway, this was a good way of validating the predictions. So to summarize, then, I hope I've given you I've gone through a lot of complex topics, I guess, in a relatively short amount of time. I've shown how we can use automated binning to get MAGs directly out of metagenomes, and the underlying principles there are basically some form of data transformation followed by a clustering algorithm. And then I've shown how we can use those MAGs to understand community dynamics, and I talked about trait inference to help make sense of this diversity. And then I showed how within those MAGs, you can actually resolve subpopulations using the time series information. And with these graph-based methods, the ones working directly on the assembly graph, we can actually get pretty reliable predictions of haplotypes. And yeah, suggests intraspecies niche partitioning in both the anaerobic digesters and the oceans. And yeah, in actual fact, what I'm working on right now is using the same principles for finding SARS-CoV-2 lineages de novo in wastewater. So that's taking up a lot of my time at the minute. And if these areas interest you, we've actually got at the Earlham, we're recruiting at least 1 postdoc next year and a couple of PhD studentships. And I'll conclude with acknowledgements. Seb Radigu, my postdoc at the Earlham, actually did most of the bioinformatics here. I work on the algorithms. He does all the pipeline engineering. And then Orkan Sawyer helped organize the AD study. Sergey Nurk, in particular, did a lot of the graph manipulation, those extraction of subgraphs. And then Maren and Andrea and Tom all helped with providing data sets. So I'll conclude there, Alice. I don't know how I did for time.
Yeah, you're almost pretty much exactly an hour. So thank you very much.
That's not too bad, is it?
Yes, it's pretty good, and I really enjoyed your talk. So it was very nice mixture of both theory and applications. So thank you very much for that. And Yeah, I mean, also the strongest is a really cool approach, I think. I was wondering, what is your assessment of what's the coverage required to actually get a mark resolved on a strain level? And do you have a feeling about what's, how you distinguish between real variants and sequencing errors?
Okay, now that's a couple of very, very good questions, actually. In terms of the coverage required, it's a little bit counterintuitive because you might think that for MAG resolution, higher coverage is better. And that is generally true up until a point. But once you get a very high coverage on a MAG, then this strain diversity becomes a problem and the MAG will fragment more. So actually for generating MAGs, there's sort of like a sweet spot of coverage, which is enough coverage that you get assembly, which is about more than, say, 10 or 20, but not too much coverage that you start to see the strain variation if it's present. So, oftentimes we get the best MAGs between 10 and 20 coverage and maybe a couple of hundred. But surprisingly, those things that you have sort of massive coverage for that you would think would be ideal actually don't resolve very well. But for the strain resolution, there we can deal with high coverage. Obviously, that's sort of the point. Although, and there, you can resolve a strain provided the coverage is again enough to assemble, basically between minimum of 5 and 10. I guess.
I see. Yeah, that makes sense. I guess then it also depends on the limit on having like strain resolved assembly algorithms to start with, and one can get better strain resolved MAGs in the next step, right?
Yeah, no, that's a good thing to think about. So as I sort of say, the way we get the strain information is we start from the assembly without simplifying it, but you were asking about sequencing noise as well. And we do actually resolve some of that on the graph by sort of doing local estimates of coverage on the graph and then removing any node that's smaller than a certain fraction of that local coverage estimate. So that's how we deal with noise. But actually, there isn't really a strain resolve binning algorithm that's currently available. We're still using the Contigs, basically, for
Yes,
the actual MAG, for the actual binning. So we're not using the strain information in the binning process. It's something we're sort of applying as a. So actually, that's a sort of that would be a good area of research, in fact.
Yeah, I think a couple of people are working on this, and I'm not sure if you've seen. So on a related note, if you've seen the performances. So you mentioned Concoct that you've worked some time ago. So Concoct actually performed quite nicely in the second Cami challenge. So if you haven't seen those results,
it did better than in the first one. That's encouraging.
Yes, it did actually very nice on the common genomes. So because so in several settings. So that was an interesting finding.
Yeah, that's actually how I feel about Concoct. I mean, it's a very old algorithm now, but I mean, I don't think it's necessarily the best algorithm for any particular binning. But as I said, because it actually has very little assumptions in it, because everything's variational inference. I have a very weak priors on everything, so it sort of always does pretty well. Even when you're not looking for prokaryotes, maybe you're looking for eukaryotes or viruses. So I still sort of find it a useful thing, actually. Which is nice, because it is quite an old algorithm now, and there's a lot of competing binning algorithms.
That is true. It's a very busy field. Yes.
Yeah, it is. Yeah.
So, I mean, something that you also contributed to back to the first Cami challenge was metrics for assessing those binning algorithms. And so now people have converged like on a common set, like using the completeness, contamination, but then also the ARI together with how complete the sample is. So do you think that covers basically relevant metrics, or do you have other things you think that are important to look at?
Sorry, what was the other metric you mentioned? The R rate. I'm not clear on that.
The adjusted rand index.
Oh, right. Okay, so that's when you've got a gold truth in it. Yeah.
Yes.
You've got a ground truth, a gold standard in effect. Yeah. Then, No, I sort of feel that does cover it. I mean, well, there isn't. Well, up to a point. I mean, so when you're doing actual practical binning, almost certainly the only thing you're going to look at is the number of good quality genomes.
That's true. That's
so
another one.
I sort
Yes.
of feel that for the evaluation, that is the most useful metric. But the problem is, and this is something that perhaps is nicely illustrated by Cami, is that you can't trust entirely those single copy core gene predictions of completion and contamination. They're always an overestimate of how good your genome is, and in particular, those accessory, the accessory genes that I was talking about, they're most likely to be incorrectly binned, in fact. And you won't see that when you look at the completion and contamination metrics, in fact. By the way, what did you think? Since we don't have any questions from the audience, I'll ask you a question. What did you think of the trait inference? Because we're effectively doing the same thing as you did back with Traitor in
Yes.
2016, but by applying it to MAGs.
Yes, so we had a little piece on Max back then, but just a smaller one. And so what I really liked in the results that you showed was this, I think that you applied it to the whole community. So it was really nice. And that you were, right?
Yes,
So
we have done that. The results I was showing were actually just from Methanogens.
Yeah, and that you had this that you sort of made some statements of how the trait of the overall community changed, and that I thought was really interesting. So
Yeah.
that's extremely useful. Predicting
I think
traits.
so.
Yeah. So I think that's really cool work, but it's also a really challenging problem. Now, if you're looking at it, just because you have this whole diversity of strains, below the species level. And so when we're looking at this, I think for some traits it generalizes really well, while for others, I'm not so sure if it really, like for some medically relevant traits, whether you can really straightforwardly apply these models and they will be reliable, let's say, across the whole bandwidth of different E. coli that you have there, because they're so divergent on a genomic level. But it's a really fascinating, important problem.
No, that's a good point, actually, and it relates to the rest of my talk. So I should perhaps have raised it, in fact, because obviously, when we're applying the traits to MAGs, we're ignoring the accessory genomes.
Yes, that is right.
And we don't, from the strong algorithm, we don't actually have a good method yet for resolving the accessory genome. So if you've got any traits that are, sort of quite fine scale, then that's a good point where we aren't necessarily going to get them correct. I hadn't really thought about that, actually. But no, it sort of suggests why we need to go on and actually get the accessory genome as well. And perhaps you would have the resolution for some of those fine scale traits then.
Maybe that's the key to that. That's true. Yeah. So last chance to ask any questions. If we don't have one in the talk, well, there's none there yet so far, but so then I would just like to thank you again, Chris. This was really cool, and this is going to be an excellent addition to our webinar series that's available. Here's a question. Let's see, where's that?
It's posted in the Q&A, I see.
Oh, okay. So, I should read them also. So Fernando is asking, what metric would you recommend to assess binning performances? And do you still consider the adjusted round index a good metric? But that's along my question. Because
really, I didn't realize that it did well in Cami too. I hadn't looked at it. So that's nice news, actually. Well, there you go.
Yeah, that's true. And then what purity decreased mostly with increasing strain diversity? Is this? Yes. So this is something that we saw that, and is this something that you would expect in general?
Yeah, definitely. Because what's happening there is if you've got more strain diversity, then you've got these little fragments of genomes, and they're both going to be small, and they're not going to have reliable coverage profiles. Because if you've got multiple strains, then each strain hasn't got the correct overall species profile that you're trying to learn. So then the purity would be quite bad, both small contigs with noisy coverages you're going to have a problem actually getting the bins correct, in fact. So that makes sense, and comes back to what we were talking about, Alice, that it would be good to have a binner that took into account strain diversity, in fact, wouldn't it?
Yes. And then there's a final question about Concoct was one of the best binners that we assessed. In the Chemie Challenge, important observation was that completeness decreased with decreasing assembly quality. Oh, right. So on real assemblies, this is also, there was a difference in performances of assemblers on those perfect assemblies versus the real assemblies, which tended to be more fragmented. And so is that something... Yeah, so that's more like an observation. Okay, thank you very much, Fernando. Any comments on that, Chris? Is that something that you would have expected?
No, it is sort of, actually. I mean, I think it also illustrates that reality is always more difficult than your ideal scenarios, right? And I think this is a problem with bioinformatics evaluation in general, particularly when people publish their own papers. They're always using fairly idealized data sets. So it illustrates the usefulness of Chemie, in fact, that you're using properly complex examples, and then you might start to see some of these effects. So yeah, it's interesting.